Alright, y'all know the format. Paper 2. This one's called Control of Neuronal Synapse Specification by a Highly Dedicated Alternative Splicing Program. It's by Lisa Traunmuller, Andrea Gomez, T-Ming Nguyen, and Peter Scheifele. Now, this paper's a little bit old. It came out in May, but I saw it. One of my friends posted it on Facebook, so, I couldn't resist doing it. Alright, so the authors start off by mentioning that in many different subsets of neurons, alternative splicing seems to be an important mechanism for customizing the gene expression, given that we have a limited number of genetic resources in all cell types. So what this paper focuses on is a protein called SLM2. They don't tell us what it stands for, but it's an RNA binding protein, and it seems to be involved in splicing, particularly in neurons, and more specifically in the mouse hippocampus. So they study the knockout mouse SLM2, KO, total null for SLM2. Alright, so the first thing they mention is, what does this null mouse look like at the level of the hippocampus? Well, they note that there's no change in the number of dendritic spines. These are glutamatergic spines. The whole paper is sort of about glutamate, so if you'll recall, I'm not too good at neuroscience, but glutamate is most definitely an excitatory neurotransmitter. So there's no change in the number of dendritic spines. The p-value is 0.5, which is not significant, and their n is nice and high. So I went ahead and changed the light a little bit because it's very important that you see these figures. So let me just go over, there's a lot of heavy neuroscience stuff in the first figure, figure 1. You can check it out for yourself if you have the time, but I'll just go through the highlights for you. So yeah, no change in dendritic spines. Obviously SLM2 is absent, this RNA binding protein. Another interesting thing, I guess, is the miniature excitatory postsynaptic current, MEPSCs. Very hot these days in neuroscience, you've probably heard of it. Anyway, the MEPSC is a tiny bit faster in SLM2KO mice. Well, I'd show you the picture, but you can't even barely see. But the p-value is small. The p-value is less than 0.05. Okay, what else? Well, the AMPA receptor is increased by with respect to the NMDA receptor. Again, so the AMPA receptor is a glutamate receptor. Not entirely sure what the significance of that is, again, because I'm not really a neuroscientist, but the gist is there's an effect on the types of receptors that are expressed at synapses, and it's quite significant also in this SLM2KO mouse. And finally, something I find pretty interesting is they have a technique called theta burst stimulation. Basically, it's a measure of LTP, long-term potentiation, and amazingly, you can check it out, it's completely abolished. It's completely abolished in these mice. Here's the wild type, that's the KO. So, yeah, quite a reduced response to long-term potentiation there. Okay, one other really important, interesting part of this is that SLM2KO mice have actually a great increase in the GLU-A1, which is, anyway, it's just a component of the AMPA receptor. So check it out, Western blot, nice blots here. I would call that a 1.25 x increase, and that's even bigger. So the first column is actually in the synaptosome fraction. So the first column is actually in the synaptosome fraction. Not sure if you're familiar with that, but It's similar to microsomes. You know, if you do a subcellular fractionation, you get all these tiny little vesicles if you do a sucrose gradient. So synaptosomes are one kind of vesicle. The other bit is in the Triton soluble fraction. They also see this GLU-A1 is way, it's upregulated in these SLM2KOs, which means, what does this mean? It means SLM represses GLU-A1. And they checked these other receptors, NMDA receptors, and they didn't see anything different. Okay, another nice Western blot here. I'm not entirely sure what the difference is, but here again they show there's this increase in GLU-A1, about a twofold increase when they do a bit of a pull down for that. Okay, so what do? They want to characterize the expression changes in this null mouse. What do? Obviously, they make a volcano plot. Look, that's so good to see. This paper was published in Science, so you know they are good at their volcano plots. Just like to emphasize, it's very important to always make volcano plots of your high throughput data, because look, on this axis you've got the full change either up or down, so each side will be either up or down, and then on the y-axis you've got your p-value, which tells you you can't just go by the full change. You have to include this p-value too, because depending on each gene and your sampling, sometimes you think you have a hit, but it's insignificant. So in the volcano plot, you have two branches that go up like this, and you're looking for the extreme points on both axes. What do they find? Well, let me refer to the text. Well, this is kind of what's interesting in here, I guess. So genome-wide splicing is altered because this splicing, not splicing factor, but RNA binding factor is deleted. And basically what they find is in this volcano plot, basically, sorry, I didn't really explain it, but this volcano plot is basically measuring the alternative splicing. So the x-axis is going to be the fold change in a certain exon being incorporated or not in wild type or KO. So basically they find that splicing of a few genes is greatly upregulated. For example, these neurexins, which are known to have an alternatively spliced segment called AS4, alternatively spliced segment 4. And that's going to be the focus of most of this paper. Alright, so I can hear you saying already, right, volcano plot, well, theoretical, right, full change, not quantitative. So here you go, Western blot, validation of each of these hits. You can see they've got their primers here, basically going to detect if there's a large heavy band, it's going to be this alternatively spliced. If they've got the small band, right, it's going to be not containing this extra exon. So here you go, neurexin 1, 2, and 3, all there. In the wild type, the bands are about equal, so it's about half alternatively spliced. In the KO, it's always alternatively spliced. Some other hits, this STXBP5, hang on, let me see, refer to the text. Oh yeah, that's actually called tomosin 2. Yeah, so that's also alternatively spliced. The second, there's a bit in this figure, there's just a bunch of Westerns showing genes that they thought were hits but are not hits, right? And they have good control here too. Interestingly, this tomosin 5 actually switches, one of the exons basically stops being present and it switches to another exon. So it's an alternative splicing that replaces one exon instead of adding or removing. Okay, now another volcano plot. This one was a bit confusing, but basically what they do here, you know, look, it looks the same. But here they are now testing, they're doing a purification with an affinity column that has neurexin, let me get this right, that has neurexin actually on it. So neurexin was alternatively spliced. Now they're going one level further, they're finding the proteins that interact with that. And, you know, they say, oh wow, we found some interesting hits. Some of these hits I find unbelievable. So they've got neuroligin I'm not entirely sure, but the name seems to suggest that it's a kind of connective protein. You know, they got these leucine-rich rich repeat-containing proteins that are also known to organize synapses, kind of neurexin-binding protein. But some of them, look at this, they say, oh wow, it's a previously unknown candidate ligand, complement protein C3. Complement protein C3. I'm thinking that's an error, but if it's true, that's really interesting. You know, it kind of makes me think it's not an error because also pentraxin, that's another innate immune protein. Pentraxin, neuronal pentraxin, also bind to this, so kind of cool. They find a lot of differences with this alternative splicing, so they find that with alternative splicing of neurexins, you basically have different protein-protein interactions. Okay, a little more direct, they do a pull down, co-IP, check it. Neurexin, before the IP, after the IP. Here's the IP. That's the protein itself. Obviously, it shows up in the IP. This here, neuroligin 1, neuroligin 2, they showed up in the volcano plot, right? Wild type, they bind. KO, they do not bind. Alternative splicing is required for neuroligin binding to neurexin. Okay, so what do? Straight away they go and say, all right, well, we found this phenotype. Now we're going to go ahead and rescue it straight away. And, you know, I was once a geneticist a long time ago, so yeah, I really dig when people do rescue experiments. That's just the way to go. So, see that. Another Western. So here you've got your neurexin 1, right? Basically, here's the wild type. It's got both splice forms. Here is your KO. It's now only alternatively spliced. Here, well, just ignore that. Then here, you've got your KO plus you've got basically this rescue. So it's going to be KO, but it's going to be unable to be alternatively spliced. So basically, you see a return to these wild type levels. See, they've actually got, look, 2 bands at equal intensity, same as wild type. And there's also some really cool, look at these results here. Neurexin 2 and 3, just completely unexplained random pattern of bands there. I have no idea how to interpret that. I guess they didn't either. Their rescue now has functional consequences. Look, now they're going to rescue, in an IP, they're going to rescue the interaction of neurexin with its neuroligands. Look, wild type, KO rescue, wild type, KO rescue. That's pretty convincing to me. What else? They're going to rescue the expression of the AMPA receptor. So look, wild type, AMPA receptor increased. AMPA receptor is now back to normal. And obviously no change in the NMDA receptors. And they've got good controls for that too. Third thing, they've got some behavioral tests even. So they find that wild type mice like to explore novel objects. So look, I don't know what the name of this test is, but they like to explore novel objects. So there's the novel, there's the familiar. By contrast, these null mice they don't like to explore novel objects at all. They prefer familiar objects, rescued by this alternative splicing construct. Oh, and the best part about it, okay, you know, I was talking about this theta wave stimulation. That's a model of long-term potentiation. Well, basically, here's that. It looks kind of like the same experiment, right? They've basically done that again, and they've got, you know, wild type KO, and then they can partially at least rescue this. Basically proving that this LTP is definitely dependent on splicing. And not just splicing as catalyzed by this protein, SLM2, an RNA binding protein. Okay, so I know that was a bit much to take in, so let me go through it. So basically, SLM2 regulates splicing in neurons. How do they know? Because in SLM2 null, brains, they have all these differences in spliced isoforms, particularly in a protein called neurexin 1, which contains this conserved region called the alternatively spliced 4 segment, and also in other proteins called tomasin 2. Basically, they show that because of this alternative splicing, so you've got 2 possibilities. One, the gene is normally expressed. The other, it's spliced containing an extra exon. And basically what they're going to say is this splice produces a different protein which is required, that segment that's translated by that exon is required for interaction with a certain set of proteins called the neuroligands. And basically, this has effects on the arrangement of synapses, not only the establishment of the synaptic organelles, but direct functional consequences on mouse behavior and on their electrophysiological behavior too. So let's read the abstract. Alternative RNA splicing represents a central mechanism for expanding the coding power of genomes. See what I said. Individual RNA binding proteins can control alternative splicing choices in hundreds of RNA transcripts, tuning amounts and functions of large numbers of cellular proteins. We found that the RNA protein SLM2 is essential for functional specification of glutamatergic synapses in the mouse hippocampus. So yeah, I might have skipped that a bit, but basically, yeah, all these proteins are basically involved in recruiting, organizing, and developing the synaptic organelle at a certain part of the cell. A genome-wide mapping revealed a markedly selective SLM2-dependent splicing program, primarily consisting of only a few target messenger RNAs that encode synaptic proteins. Genetic correction of a single SLM2-dependent target exon and the synaptic recognition molecule neurexin-1 was sufficient to rescue synaptic plasticity and behavioral defects in SLM2 knockout mice. These findings uncover a highly selective alternative splicing program that specifies synaptic properties in the central nervous system. That's all the time we have for this week. Thanks for watching. Please go check out these articles and like and subscribe. Bye.